Every metabolic process inside a living organism extracts energy, and in doing so, produces a lethal byproduct, nitrogenous waste. If these cellular toxins accumulate, the organism dies. For a single-celled organism like an amoeba, disposal is a matter of simple geometry. Because its entire outer surface is in constant contact with an aquatic environment, toxic byproducts diffuse directly outward into the water. Complex, multicellular eukaryotic life faces an architectural crisis. Trillions of internal cells are trapped deep inside a closed biological system, completely isolated from the external environment. This creates a mechanical problem. The organism must forcefully filter and expel cellular toxins from its internal fluids, without accidentally draining the vital water and nutrients necessary for survival. Complex life solves this biological contradiction by scaling up a precise mathematical principle, the semi-permeable membrane, taking it from the microscopic surface of a single cell and building a massive internal filtration engine. The discovery of these boundaries began in 1665. Using rudimentary optics, Robert Hooke observed thin slices of cork and identified what he called cells. Because the cork was dead, what Hooke actually saw were only the empty, rigid outer boundaries left behind by the plant matter. It required the improved lenses of Antony van Leeuwenhoek to look into pond water and identified the first active, free-living bacteria swimming in the fluid. This observation proved that biological boundaries are living, dynamic barriers. They regulate their internal chemistry by physically interacting with their environment. To understand the mechanics of how a human organ filters blood, we have to first look at the microscopic architecture of these living barriers. This 3D cross-section illustrates the Singer-Nicholson fluid mosaic model. Proposed in 1972, it remains the definitive blueprint for biological filtration at the cellular level. The foundation is a phospholipid bilayer. It operates as a continuous, fluid barrier that physically blocks large or electrically charged molecules from crossing into or out of the cell. Embedded within this fluid layer are intrinsic and extrinsic proteins. They act as highly specific gates, verifying the chemical shape of approaching nutrients and granting passage only to the exact molecules the cell requires. This selective permeability allows the cell to manipulate its internal environment, discriminating between similar molecules based on charge and molecular weight. The human kidney operates on this exact biological logic. It takes the selective capability of the cell membrane and scales it up to process liters of blood continuously. While multiple organs deal with waste, their specific burdens differ. The digestive system processes undigested solid matter left over from consumption. It does not handle the waste produced by cellular metabolism. The respiratory system also acts as an exit point, but the lungs are exclusively engineered to expel a single gaseous byproduct, carbon dioxide. The excretory system has a distinctly specialized purpose. It extracts dissolved nitrogenous wastes, specifically ammonia, urea, and uric acid, directly out of the circulating bloodstream. Pulling dissolved toxins out of liquid blood without removing the liquid itself is a highly specific chemical task, demanding an entirely distinct organ architecture. Looking at the gross anatomy of the excretory system, we can trace the path of unfiltered, metabolic waste-heavy blood as it flows from the heart down into the renal artery. The filtration relies on 4 macroscopic components. The blood enters the kidneys, the waste liquid travels down the ureters, it pools in the urinary bladder, and is finally expelled through the urethra.
The kidneys themselves sit laterally in the abdomen, rather than in the anterior cavity. This specific positioning explains why referred pain from renal distress, like kidney stones, typically presents in the lower back. Once the waste is separated, the purified blood exits the kidney via the renal vein, feeding directly back into the inferior vena cava to recirculate. The remaining liquid waste, urine, gets its characteristic pale yellow color from a specific pigment called urochrome, a byproduct of breaking down red blood cells. Structurally, the kidney functions as an inline biological sieve, plumbed directly into the main cardiovascular circuit. The actual work of filtration doesn't happen at the organ level. It happens inside the nephron, the fundamental unit of the kidney. This cross-section shows the primary site of filtration, Bowman's capsule, which encases a tightly coiled capillary bed known as the glomerulus. Intense cardiovascular pressure forces fluid, salts, glucose, and urea out through the capillary walls. The pores are sized precisely so that larger red blood cells and complex proteins are trapped inside the vessel and kept in circulation. This pressure-driven mechanism is managed by antidiuretic hormone, or ADH, secreted by the pituitary gland. ADH commands the nephron to reabsorb the necessary water back into the bloodstream, preventing massive dehydration. Substances like alcohol actively inhibit ADH production, which shuts down this reabsorption process and leads to rapid fluid loss. The nephron achieves systemic homeostasis by perfectly balancing the mechanical force of blood pressure with precise hormonal feedback. When this biological boundary breaks down, the failure cascades rapidly. Renal failure results in the immediate toxic accumulation of ammonia and urea in the bloodstream. Without the ability to regulate water volume, the body develops edema. This presents as visible systemic swelling, severe hypertension, and fluid retention in the extremities. Localized chemical failures also occur. When dietary oxalic acid reacts with high concentrations of calcium, they form jagged, obstructive calcium oxalate crystals, clinically known as kidney stones. When the biological system ceases entirely, the final medical intervention is hemodialysis, a mechanical backup for blood filtration. Hemodialysis routes a patient's blood out of the body and through an artificial semi-permeable membrane. The machine executes the exact selective permeability of the nephron. Dialysis provides a mechanical bridge, substituting an industrial membrane to maintain the chemical equilibrium that the biological system can no longer sustain. Stepping back from human anatomy, comparative biology reveals that an organism's surrounding environment strictly dictates its filtration strategy. Most aquatic life forms, like fish, excrete highly toxic ammonia directly into the water, relying on the immense volume of the ocean for instant dilution. But terrestrial mammals cannot afford this high toxicity. We expend massive amounts of biological energy to convert ammonia into a less harmful compound, urea, which can be safely stored in the bladder before release. Birds and reptiles prioritize their vital water retention by converting liquid waste into a completely solid uric acid. This metabolic pathway allows them to excrete toxins while bypassing the heavy fluid loss required for liquid urine. From the microscopic gates embedded in a single cell, to the managed pressure in the kidney, to the diverse strategies of separate species, survival relies entirely on the functional